Good evening, everyone. We are here for the last day session, the fourth day of this international webinar, which has been a great success by the support of all of you. And today we are having Dr. Ratnesh Jain, who will be addressing us. We are having with us two moderators, Dr. Madhur Kulkarni and Dr. Suvarna Ingre. Now I request our principal, Dr. Anagha ma'am, to kindly address the audience.
Yeah. Good afternoon, everybody. On behalf of Sri Chanakya Education Society's Indira College of Pharmacy, I welcome you all for today's fourth webinar in the International Webinar Series. A warm welcome to you, Dr. Ratnesh Jain, sir. And we are eager to listen to you about the monoclonal antibodies, the new way of therapeutics. And Yes, I think till today, all these seminars were a great value addition to all of us, and I know that today's is also going to be the value added knowledge sharing with us. And I appeal to all the participants to get maximum from today's seminar. Welcome you all once again.
Thank you, ma'am. Now I request Dr. Suvarna Ingre to kindly introduce our today's speaker.
Thank you, sir. Good afternoon, one and all. Once again, I welcome you all for the fourth day of ICP International Webinar Series on Biologics, the Future of Therapeutics and Healthcare Management. It's a great pleasure for me to introduce our today's speaker, Dr. Ratnesh Jain, who is going to talk to us about monoclonal antibodies, the next generation therapeutics. Dr. Ratnesh Jain is an assistant professor in engineering sciences and also Ram Swami Fellow at Department of Chemical Engineering, Institute of Chemical Technology, Mumbai. He is heading Biologics Characterization Lab at ICT. From 2012, Dr. Jain has taken various initiatives towards biosimilar characterization and educational initiative like Biosimilar Workshop to support biopharmaceutical sectors related to skill development, infrastructure development, and entrepreneurship. He has more than 79 publications and more than 200 presentations to his credit. He is consultant for various biopharma industries and is also advising and assisting various biopharma startups for innovative solutions. With this brief introduction, I hand over this session to Dr. Jain. Over to you, sir.
Thank you. So thank you, first of all, to the organizers for inviting me to this webinar. Thank you specifically, Madhu, for, courtesy towards, calling me here amongst the great speaker which we have in, earlier days. I'll be starting today about monoclonal antibodies, which is, one of the largest within the, all the biopharmaceutical which are at the moment in market. These maps are specifically, created for a targeted therapy in the beginning. Considering the huge amount of complexity available for these maps, it was always a challenge to develop them. These maps are typically more closer to molecular biologists or biotechnologists in the beginning. Eventually, when they become a therapy, it was more of a consortium of different scientists who could actually now develop them and can take to the patients. So to start with, this is just a disclaimer from my side. So this particular presentation is only for teaching and educational purpose. There is no other, interpretation one should take out of this particular presentation. I'll be briefly talking about, considering a little bit delay in the presentation, we'll be, taking a little faster approach, though we have some time, but I'll just take it a little faster. So we'll be talking about ICT, and then we talk about what biopharmaceuticals are, what are the maps, how these maps are produced, how maps are characterized, how one should check the biological functionality, how to take a regulatory approval so it can reach to the patient, and finally a conclusion and outlook for my presentation. Though most of you might be aware about ICT, but ICT, just to give an introduction about ICT, it's set up in 1933. It's primarily set up for Indian chemical industry that time. It is one of the elite institution in the state of Maharashtra, as well as we have recently been ranked 92nd in the Times Higher Education ranking, and we are the top seventh institution in country by Times Higher Education, and also amongst 500 uh, uh, institutions all over the world. So this is very, and educational world has also ranked us second in Maharashtra and again ninth overall in country. So we are very proud of our achievement, which is also a very big responsibility amongst all the faculty members and all the stakeholders to continuously strive for the best performance from our side. We have some of the best chemical engineering, pharmaceutical science, and biotechnology programs. We also have very high and parallel impact to Indian society through government and as well as through entrepreneurship. These are some of the names which are our alumni and which typically controls the major share of economy, Indian economy, as well as a major share into the global economy. So these are also some of the famous names which gives a bigger responsibility for all of us to perform much better. Coming back to our group, so we are called Nano Medicine Research Group. There are two labs. One is Farm Biotech Lab, another is called Biological Characterization Lab, which is primarily handled by myself. Again, coming to biopharmaceuticals, so biopharmaceuticals are a class of multiple drug molecules. Now, these drug molecules ranges from oligos, peptides, cytokines, recombinant proteins, fusion proteins. I'm also going the way they have appeared in different literature as well as into the commercial market. Then recombinant proteins, fusion antibodies, bispecific antibodies, then monoclonal antibodies. If you see the sequence from left to right, oligos were the one which came into the market for certain treatment. Peptide came. Peptides are still one of the biggest, blockbuster within the market, and one of the nicest therapeutic regimen, which many of the patients are getting benefited. Then came recombinant protein like EPO, which was very much needed for different cancer patients. Then MAPS and bispecific antibody, who were the primary candidate, which came for different treatment option. And then antibody drug conjugate was designed specifically whether the action of drug can be combined with antibodies. So that was then the DNA, specifically DNA, siRNA, and miRNA based therapies, viral vectors, which are still in considerable amount of research as well as clinical trials are going on. Not only as a molecule for therapy, but also delivery vehicles. Then gene therapy and cell therapies, which is one of the very famous therapy which we call CAR T therapy, is amongst that category. And there are many gene therapies are still in clinical trial as well as an R&D pipelines. Now, when we talk about biopharmaceutical level, the obvious question comes: why are we talking about what is there? So one of the major driver and one of the major motivation to see these molecules, because these molecules are driving growth. And this is a next 5 years growth plan which has been proposed, and it sees that in multiple ranges from antivirals, rheumatic, anti-diabetics, vaccines, or immunosuppressant, or multiple MS therapy. So it ranges from one region to another region, and that is actually showing a very strong amount of a growth. So when we again talk about biopharmaceuticals, this is one of the major thing that these biopharmaceuticals are not very new. They are coming, they came into the market back in 1975, and that was the first mAb approved. There are multiple maps, eventually came, with a lot of reluctance, with a lot of also FDA scrutiny. But one of the map which actually, there are two maps I would say, which have changed the overall market. The first one was rituximab, which you see in 1997 here itself, and the trastuzumab, which is, anti-HER2, specifically came for breast cancer. And the success of these two antibodies, and of course the third, which came is Humira or adalimumab, which is also one of the largest selling drug as of today. It amounts for almost a couple of $1 billion, and this is the largest selling drug overall even in the market. So, but the popularity, or I would say the glamour to antibody therapy, has come from Herceptin, or which you also call trastuzumab, specifically for breast cancer. And that particular antibody and success of that antibody actually, uh, uh, you know, has created a momentum of discoveries all around the globe, as well as different companies who are primarily developing such therapies for different diseases. This is just a example. We have more than 500 antibodies. This is one of the example which you could see that these are so starting from rituximab, which is 1997. We have different kind of approved indication and format, which I'll be explaining you what the format means, and having different targets. So these antibodies specifically target specific kind of receptors, and these receptors, which in turn is responsible for the disease pattern. So these disease patterns are either cured or the treatment options are given because antibodies, and we probably know IgG or antibody from our very beginning. These are specific in nature, and that is the reason they are so specific that they create a much better therapeutic impact when they are being administered. Again, if you see approval, which is in 2009 drugs, so the approvals is extremely high. It is considerably growing. So these are the numbers which are, more of the new medicines which are being approved every year by US FDA. These are again a share. So if you could see that certain protein and oligos which are being approved, if you see protein which is in pink, which consists of 21%, which also has nanobody, fusion proteins, ADCs, MAPs, as well as we are also seeing some other approval like antisense and siRNA. So these biological molecules are getting bigger and bigger share with each passing year, and I'm sure that biologics drugs will have much more share of approvals in years to come. Again, if you see again a very very focused market opportunity, then this is the number which I was talking previously. These are certain maps which are being approved, and that is the forecast, the amount of money they are going to raise from the market. So these are also expensive therapy, but a therapy where no other therapeutics are actually working with that kind of effectiveness. Whenever we talk about biologics, major question comes, what is biosimilars and what is biologics? So when if you recall that there is a monoclonal antibody, so we are very well aware about generic drugs. So whenever a patented molecules which get uh, expired, or which basically after the patent, if somebody wish to develop that same molecule, people are free to develop those molecules as long as they are showing effectiveness. Typically, that effectiveness in the beginning was more of a clinical effectiveness, considering the lower complexity in the molecular structure that eventually went to a bioequivalence parameter. So bioequivalence was something which is right now available for generic manufacturing. When we talk about biologics or antibody manufacturing, we are still considering the complexity in the molecule. We have to have a similar, so these are called biosimilar because they are not same. A generic drug, so a benzene to benzene structure can be the same, but a protein to a protein structure cannot be same. It shows a different kind of behavior as well as considerable amount of differences, and that differences will always be with some percentage, and that's why we don't call same but we call similar. So, and that's the name which called, so any biological molecule, if you make a generic version of that, called biosimilars. Now these are also very very economic, considerable. Consider the development cost is much less, but it's still much more expensive. So if you compare that, around $1 to $5 million is a cost of development for generic. Somebody has to pay around $100 to $200 million in the biosimilar development. A generic can manufacture in 3 to 5 years, but biosimilar will need 8 to 10 years of the development timeline. And this is something. So these are again, more parameters. So chemically, these are much more less complex. Small molecule, any small molecule, say paracetamol, or any of the drug which you can take a small molecule. And biologics, which are large molecule, have a very complex production process, which I'm going to tell you today. These products are very robust, the chemical products, and you can have a tablet, capsule, multiple dosage form. Biologics molecule, unfortunately, so far we are only able to give by injections, and oral route is not something. One of the peptide, which is called semaglutide, recently been approved by Novo Nordisk. Recently been approved, is the only protein which is right now being given by oral route. But proteins typically is very susceptible because of the degradation process variability. So if you see left hand side, the process is very simple, the synthesis is very simple, the risk is low. In biologics, everything is very complex and the risk is high. But when I say risk means, so there are manufacturing risk, but at the same time the risk to the patient. And that is the highest amount of risk when somebody has to consider a risk. So when patient safety is considered, that is the highest amount of risk which is there because of the biologics molecule. And that is the primary reason these biologics molecule takes a huge amount of time for the development as well as to the approval. This is a very simple diagram, therapeutic biologics development. And it starts from, so if you see from left hand side, which is molecular clones, if you see the cursor, and it starts from there, it goes to process development, analytical development, biological activity, pharmacology, toxicology, and then clinical. So this is the total activities. There is nothing beyond this. Everything is consistent. So if one has to develop a biologic or biosimilar molecule, it has to be started from the clones and it has to go to the clinical. I'll be today focusing on clones, little bit about process, analytical and biological activity, and we'll rush it to regulatory, which consists pharmacology, toxicology, and clinical. Again, another slide to tell you that how this is different. So these are the small molecules, the simple aspirin, and this is, a lot of people compare by different routes. You can compare that this is bicycle and this is Titanic. So the complexity between a bicycle and a Titanic, whatever is there, the same complexity you can see between a small molecule and a protein. So what are the monoclonal antibodies? Monoclonal antibody is a simple IgG structure, typically in the Y shape. So one can see that, how it is actually seen. This is typically called hinge region, which is attachment region, where all the sites are attached. This is something which is called, antigen binding site, if you see this particular part. And this is how any antigen will actually bind. The V correspond to variable regions. So these are the different regions. L is light chain and H is heavy chain. So variable and constant region, C for constant, V for variable. So these are the light chain sequence and these are the heavy chain sequence. So heavy chain sequence and light chain sequences are going to comprise primarily a antigen binding site. Everything here is attached between these two regions or these two regions or these two regions. Everything is attached through a disulfide bond. It's very important to understand these bonds. And end of the day, everything goes back to the chemistry. There are certain carbohydrates, which we also call glycans, are attached. These glycans give stability to this structure. This is a very large structure. As I'm talking about, somebody can compare that it's not, 3.5 kDa, or it's pretty large structure. So insulin is, you can consider that something like 3.5 kDa, and adalimumab molecule something like 45 to 50 kDa. So this is the difference of a molecule. So it's a very large molecule, and this is something which is responsible, effector region, which is responsible for biological functionality. So this is about antibody. One antibody can look. There would be typical variability happens here in the variable regions, and this is how antibody is looking like. There are many terminology which you have seen in earlier slide also, that what is chimeric, what is murine. So there are multiple. Initially, all the antibodies are typically came from murine origin. The injection has been given to a murine population, and then antibody was expressed and collected and further purified and characterized. And this is how antibody discovery started. Eventually, it was realized that any antibody, specifically murine antibody, when it was given to a human patient, there was a different kind of a response which we have got, and I won't say this is a desirable response. It was a undesirable or side effect kind of a response which has been got, and that has allowed discovery of some more antibodies which we call chimeric antibody or humanized. Chimeric is, since a conversion from murine to humanized, this was not easy. People started with certain different kind of a mixture. So chimeric is a murine and a human mixture. This is a human which is D, and A is murine. The combination of this actually makes a chimeric antibody. Infliximab, either multiple mag, rituximab, which one of the first mab which came, and which is very very well established in clinic as well, is combination which allow elimination of many of these undesirable effects. Further, the conversion took place where only certain sequence of murine antibody has been taken, but largely it was a human sequence. So when the human sequence were making these antibodies, these are called humanized, but these are not completely human. Trastuzumab was one of the humanized antibody, which has also shown extremely nice or extremely powerful clinical performance. And that has also validated that humanized antibodies are also equally powerful in terms of performance. And then human antibody, completely human antibody, which are right now possible, and they also possess high amount of specificity as well as effective functions. These are monoclonal antibodies which are available. So this is fragment. Any antibody, one can actually uh, do multiple actions. So if one has to fragment, understand what is going on, you could actually see it here. So this is a cleavage site. Every site where these Y-shaped acids, so one can cleave. Multiple enzymes are there. Papain is just for example, but there are multiple enzymes which can actually provide such sequence here, and this bond can be broken by multiple enzymes. So this is, both are possible. You could break it here and you go up peptidization, where the FC fragment and a FF fragment. So FC fragment can be, this is the FC fragment, and this is the FF fragment, which can be broken here by peptidization. You can also break. And whenever one has to characterize, one has to understand the function of antibody, you have to actually digest this antibody, because it is such a big molecule that you cannot, understand it while seeing as a full molecule. You have to always fragment it, and in the fragmentation, you could actually understand this molecule much better. There's one more question people ask: what is polyclonal? What is monoclonal? Polyclonal is, our blood cells typically produce multiple antibodies. This is a mixture of cell produce mixture of antibody. So when the antibodies does not have any specificity, not a single antibody or mixture of antibody, and they react with various epitope in a given region, then only you can call it a polyclonal antibody. Monoclonal antibody specifically, attached with a specific epitope. like in HER2 receptors. Only one epitope has been dedicated, and these are typically developed by a single quality of the cell. So this is how monoclonal and polyclonal antibody differs from each other. Now the question comes how these antibodies are produced. So there are, historically, as I say, murine mouse hybridoma. That was the first that immunization was done with, a specific kind of antigen. So it basically generate those antibodies, and then the cells are isolated, which you typically hybridomas. You screen them out, and you get a mouse map. Now, mouse map can have multiple possibility. When do a CDR graft, you can do humanized maps or a chimerization, which also chimeric maps, which I have just explained it to you. There is another technology which we call phage display. Phage display is basically based on the bacteriophages, which you know very well aware. And then these bacteriophages also, specifically attached to a very specific kind of a map. Again, you could screen them out, figure out which one is finally, working, and then one can construct the human IgG. So the second phage display actually gives, a very, very nice example of human maps. Transgenic mouse, mouse which is genetically modified already, you can also immunize the same way, get the hybridoma same way, screen them out. But in that process, you get a human map because the particular sequence of gene is already been silenced. You can also get it from B cell. B cell is always one of the biggest source. The only thing is they secrete a huge amount of antibodies, and monoclonal nature is not maintained. You could always label it and get a PCR construct ready from the human body. So if you see the current corona case, that is the same thing happening, where these recovered patient blood is being isolated, and everybody is trying to see which antibodies are there. And if they find antibody, they are going to construct through PCR, and basically will be able to design the map. So this is how this particular approach is done. But before this particular thing is done commercially, it is not a realistic technique. So commercially, you have to go to a very different approach. So commercially, a couple of cells are being. So initially, everything has been done in hybridomas and blood cells, but these are not viable option. The primary reason is, industrial work happens in industrial settings. An industrial setting allow operation of bioreactors, operation of different kind of a condition which is otherwise biologically not possible. Any blood cell or any cell which is coming from a human origin have its own sensitivity. So these cell does not survive in an external atmosphere. What had happened that then scientists were looking to different cell types, and they realized that CHO cells or Chinese hamster ovary cells are very well can be taken into consideration. One of the primary reason for Chinese hamster ovary was they didn't secrete anything when they were chosen. It means there was a non-secretory cell. Though you need a secretory cell who can secrete the antibody of interest, but then initially they were a non-secretory cell. And that was the reason they have been chosen, because they were very rugged and sturdy. They can be cultivated anywhere. The cultivation was also very critical. So these Chinese hamster ovary cells were genetically modified, and they were modified so they can secrete only one protein. And that is also one of the reasons these cells are selected majority as an industrial workhorse, because these cells only produce the target protein. So separation and further purification becomes much easier for any process to take care. What happens typically is a process called transfection. In a transfection, a target genetic sequence is typically sent it through a plasmid as a vector to a bacteria, and then bacteria basically produce multiple clones of that particular sequence which is being transfected. And these clone DNAs are then further transfected into target CHO cell, and CHO cell then carries the gene. And when these genes are multiplied into multiple population, it means if you have a 1 million population of a cell, almost 99% of that population should be able to carry the gene which has been inserted in the beginning. and that gene is actually carrying which protein has to be synthesized or which maps has to be synthesized. And then one has to isolate the single cell. The single cell isolation always becomes a very tough task. So single cell isolation, because single cell will carry the pure antibody sequence, and then single cell has to make its own copies. So when single cell makes own copies, it becomes monoclonal, and that's why we call monoclonal. Why it's monoclonal? Because it's coming from the single cell. And then clone development process. So whatever clone which is now being produced from single cell, you have to develop so it can actually be sustainable for over the period of time. And then that clone is characterized into the flask as well as in a fermenter or in a bioreactor. And that's how the development process begins. That's not the end, that's just a beginning. What happens when these transfection, as I told you, and then the cell pools are typically generated in a 96 well plate or 384 or 2000 cell, depending upon the cell quality. And then what we do is we allow them to grow for continuous periods. So if you see the timelines, cell pool typically takes 4 weeks. Cell pool expansion, it means you are allowing those cells to grow for a more period of time. When these cells are grown and they are consistently producing the protein of interest, then only these are called cell clones, which are the pool expansion which is happening. And once these are 100% grown, producing right kind of protein, they have been gone for banking, and that the process is called cell banking, and the whole thing is called master cell banking. When the master cell banking, basically is being processed, at the same time, clone generation. So the clone which is coming from the cell uh, is being shortlisted here. These clones are then further converted to clone expansion. We will again go for a cell banking, and that's typically this is the master cell bank. This is again a master cell bank here, and then also a working cell bank. So these working cell banks typically produce protein of interest when the antibodies are being produced. The total manufacturing process looks like this. So initially from a cell culture, it goes to a flask, it goes to inoculum preparation, and then through a bioreactor process, we are actually harvesting the cell. Once harvesting process, which is typically done by centrifugation or filtration, you get an antibody out. And these antibodies, either through precipitation, extraction, there are multiple steps, can be removed from the supernatant. And then multiple chromatography state, which we call cation exchange chromatography and anion exchange chromatography. So these cation and ion exchange chromatography steps allow purification process. Since the entire cell lines are working in an open atmosphere, there is a likely chance of either inbuilt viral infection or in-process viral infection. But viral inactivation is a primary part before these goes for a polishing chromatography. So that is typically done by low pH or high temperature. And then the further viral filtration step, where through an appropriate filtration mechanism, viruses are being removed. All the activities which are done, chromatography, precipitation, is typically for any other impurity now which is coming from the cell is for that particular purpose. When the final, after diafiltration, you receive an antibody. This particular antibody you call drug substance or DS. So this DS is then further formulated. Now DS is simply an antibody like a drug. It doesn't have the delivery efficiency. So then to make it stable and to make it deliverables through certain mechanism, you need to add buffer, salt or protectant. Every protein is typically, as we know, these are zwitterionic material. So they are specifically only stable in a specific pH condition or pKa condition. And that is typically provided by protectant and buffers and salt. They are also complex molecules. So one of the primary reason that they aggregate very fast. So that is also one of the reason we have to use a lot of protectant material here. One could lyophilize them, and after pure quality control and packaging, one vial could be ready here. which you could see. So this is how a monoclonal antibody injection vial produced from the entire mechanism. Now, when we have understood that how these are produced through cells and through a detailed process, one has to understand how these are then characterized. One should understand there are multiple characterization. So always understand a map is like an elephant, and the elephant which is seen by the seven blind elephant, if you know the story. So these blinds are actually trying to understand the elephant, and the blinds, when they are touching elephant, they will be explaining in their own language, which is not elephant but which is just part of the elephant. So that is the same problem happens in there is no methodology, there is no analytical methodology as such which can in one go understand these antibodies and evaluate them like a small molecule. So if you do UV characterization, IR characterization, NMR, mass, or HPLC, you understand the molecule properties. But one analysis will not be sufficient here because only one part of the antibody will be understood. So there are molecular characterization which are very, very important. There are functional assessment. In molecular characterization, proteins primary structure through typically done through mass spectroscopy. Then we have a secondary structure. We are very well aware about that protein behaves and differently. It has having a primary structure, secondary structure, tertiary structure, and based on the condition, they form different kind of a structure. So the secondary structure of protein can also be by some biophysical method called circular dichroism or FTIR. Then we also understand this structure, which is called higher order structure. Now, higher order structure is a tertiary structure and beyond. Now, when protein is folded into a specific kind of a, architecture, that particular architecture is very, very difficult to understand. So then that's called higher order structure, which can be done again by hydrogen deuterium exchange, mass spectroscopy, XRD, fluorescence, biophysical techniques like light scattering, analytical ultracentrifugation, many techniques. Then one of the very important part comes into the picture, which we call glycosylation. Glycosylation means the carbohydrate molecule. If you remember the antibody structure, these were attached. These carbohydrates are very, very responsible for their biological as well as stability. Now, understanding them, carbohydrate sequence is very, very important. This is typically done by capillary electrophoresis, HPLC, or mass spectroscopy. So this carries a huge amount of molecular characterization work on the left-hand side. On the right-hand side, we have functional assessment, though it is important to understand whether the protein is right or not right. But even if it is protein is right, whether it's functional, it is giving biological function or not. And that is typically done by different kind of assays, primarily ELISA. ELISA is the first method typically allow to understand whether ELISA is simply antigen binding assay. So we see that. There are more focused assay, which will basically say the potency of antibody, how much dose is sufficient for a specific biological action. And that is called ligand binding assays or in vitro potency assays. We also have some competitive ELISA and BIA-core, which is based on a surface plasmon resonance. And then finally, we have a cell-based assay. Cell-based assay these are the natural cell or engineered cell, which will be basically creating the function these antibodies are created for. So suppose those antibodies are anti-HER2, then these cell-based assay will have that receptor, the HER2 receptor. And basically the binding constant, binding ability, and response. If HER2 receptor binding is happening, then definitely some biological response is going to be generated by antibody attachment. So measuring that response requires a cell-based assay. And that is something which is typically done through functional assessment. We also have effector functions, which are through ADCC, ADCP, and CDC assay, which is called antigen-dependent cellular cytotoxicity. So these are other features which I'll be, explaining in the later slides. So since there are so many parameters associated, multiple testing methodology has to be adopted. And each of these parameters is called critical quality attributes. A critical quality or CQA of a protein molecule or a recombinant protein or NM. and every attribute has to be tested by a different. So typical manufacturer, normally see around 100 attributes and more than 40 assays. So such a length of experiment one has to do if you are developing these molecules. So because of a certain kind of structural variability and a heavy structure, it goes for aggregation just to minimize energy. So one should understand how to measure these aggregation. Then disulfide bond, which is a very, very critical bond which binds everything. So one has to, of course, measure the disulfide. Fragmentation, which is happening from the hinge region. Deamidation and oxidation, or any truncation, which is multiple amino acid which are there and which can be modified. And then glycosylation, it means specifically sugar molecule. So how do you do that? Because of SAC, HILIC, RP, IEC, or reverse phase chromatography. So there are multiple chromatography methods are there which can be utilized, and these chromatography methods will allow measurement of these attribute. In addition, mass spectroscopy is typically very, very important here, which also allow most of these critical quality attributes by mass spectroscopy. So this is, there is another word which is, coming, is called post-translational modification. I'll just explain it here. So these proteins are typically manufactured by cells. Cell have a process called transcription and translation. Transcription is a process when DNA is converted into RNA. And when RNA is converted into protein, that's called translation. After translation, a protein comes in Golgi bodies. And when it comes to Golgi bodies, it is typically decorated and polished. And it is also it's a synthesis process, a biosynthesis process. So anything which is not needed in that protein is typically removed in the Golgi bodies. So those modifications are called post-translational modification. And we should be able to, understand and test all these PTM. So LCMS becomes a very important tool to test these methods. Again, I'll tell you again, showing you the same method, how it is manufactured. So multiple analysis problems comes when so the first process where from cell, you are trying to develop a bioreacting process in bioreactor, which is called upstream bioprocessing. Here, there are three very important analyses to be done. One is called aggregates, another is called glycan, it means carbohydrate, and purity and yield of the antibodies. Second parameter, which is typically come downstream manufacturing or downstream analytical process. Here again, aggregate becomes a very critical task. SCP is host cell protein. So many of these host cells, HO cell, also release the inbuilt protein, which is called protein, host cell protein. That host cell protein may interfere with our target antibody. So the host cell protein has to be removed from and that removal process, we have to utilize multiple downstream steps. Protein A is basically a affinity-based column. So affinity chromatography is the most successful chromatography till date, where target protein of interest is isolated by protein A. So protein A also sometimes leach out and comes into the process, and that comes as an impurity. So this impurity has to be again removed from a downstream analytical process. Purity yield again becomes an analytical task and handle. This is typically, so these two step upstream and downstream are more of a DS stage, which is called drug substances. Now the formulation development is a more of a drug product. And the drug product again aggregates becomes a major issue here, and secondary structure and also higher order structure. Now, why these two things comes into the picture? Because proteins are now being mixed with buffers, salts, and protectant. So as soon as other molecules are interacting, so molecule to molecule interaction allow change in the structure, or it may happen, and that's why it's important to check secondary or higher order structure. Again, purity of the molecule becomes very key requirement here, which needs to be checked. This is more of a in a product consideration, that when products. So global means a global information what is needed. it's very important that one basically understand mass, shape, solution, and charge of the protein. Then what we need again a tertiary and secondary structure, which is also measured by multiple methods. There are many other methodology which will be utilized for glycosylation, sequence variant. Each protein is not going to be the same. So whatever the protein sequence coming, there would be certain variation. So these variation has to be calculated and quantified. Post-translational modification, which is called PTM, and degradation. This also require multiple methodologies to be utilized. We also have molecular characterization, which I told you in the beginning, that function, size, charge, identity, pH concentration needs to be checked. And drug product system, we also require excipient. So any residue metals or degradation, which can be happening through check by ICP-MS or ELSD. And even packaging becomes very, very critical here. These are typically packaged and ready to use injections. So these injections, these are typically comes with the drug molecule. And sometimes there could be a leachable, or there could be extraction, there could be metal lines or glass or organic. And multiple things are possible. So these packaging becomes very, very critical, because even a small amount of leachable coming out from a injection can create a huge amount of immune response. Since these are injections, immune response becomes a very critical part to be checked. And that is the reason you need to be very careful while analysis. Again, once analysis has been done, the question comes how to check these biological functionalities. So as I told you before, we need ligand binding assay as well as cell-based assay. So the first test is typically done by ELISA. We have multiple ELISA kind of a test, format A, B, C, D. Each differs from the other one. Either it's a direct ELISA or a competitive ELISA, or where we are using a specific kind of a tagging pattern or a label binding molecule. So these are, competitive ELISA of BC. BC is specifically considerable amount of the time they are being used, and D is also used, but more of on the expensive side. Then we also do cell-based bioassays, which is typically seeing whether the antibody actually interact with the receptor. And if it does interact, what happens after interaction, and measurement of those cytokines or inflammatory. And this is typically done on an engineered cell or a cell of a choice. And then once these two results comes, we do a more parallelism testing, that how the response can be compared between ELISA, which is very antigen specific, cell-based, which is more real-time biological functionality. Again, effector function, one has to do multiple testing, ADCC, CDC, and ADCP assays. These are the three different kind of assays. This can also be done by natural cell as well as into the engineered cell. These are typically to understand, the effector function, whether specific kind of, biological functionality is being given by the target antibody, or if it is not given, what are the toxicity related issues which needs to be investigated in the effector function. And at the end of the day, this is typically created for certain actions, so they have a target effect. So these assays are typically designed so they will actually express the target effect. So suppose the target effect is a breast cancer, of course the cancer cell death is the end of the target effect. So what we are going to see here, whether the cell death is happening, if it is happening, at what rate it is happening, and whether it is also not creating any other death into the natural cells which are available. To understand regulatory approval process, so regulatory approval process, this is a comparative. All the small molecule nowadays go to 505B2 or 505J route, and these are the drug and generic molecule which is being approved. When you see, biologics, so typical biologics happens through full safety and efficacy investigation and all the essential investigation, then only biologics get approved. But 351K, which is the route designed by FDA, here it is only, typically there are two
It's
ways.
5 o'clock.
It is typically done then the molecule is highly similar, and then only you can take this route. And also you have to demonstrate clinically meaningful differences here. what are the differences which are clinically meaningful, and then only you could get a biosimilar approval. One has to really see the difference. This here we really don't have to go for a clinical trial, but here you must go for clinical trial, and that is the reason that generic drugs are cheap, but biosimilars are not, uh, that affordable. The reason is there is a huge amount, the complexity of the molecule as well as the cost of the clinical trial, which does not allow manufacturer to reduce the price for any of these biologics molecules. This is a very good comparison between now biologics and biosimilar, which are biosimilars are much more economics than their biologics counterpart. But if you see the efforts, so molecular characterization to phase 3 clinical study, this is the original biologics application. But once you make biosimilar, majority of your investigation goes to physicochemical characterization and biocharacterization. If you see this blue part, this is the highest within the pyramid. It means if somebody is developing biosimilar and putting a lot of emphasis in analytical characterization, they can have much better chance for clinical success. And then you do a certain non-clinical studies and PKPD, which is more of behavioral or animal studies, and then you go for a clinical settings. This is the route which has been designed. So clinical trial is only a phase 3 clinical trial here, not the phase 1 and phase 2, and but most of the effort. So here, this particular blue part is called analytical biosimilarity, and this particular seed that called clinical biosimilarity. But if you achieve analytical biosimilarity with all possible studies, there are likelihood that you will not be failed in the clinical settings. You'll be qualified. So one has to be from the beginning has to be very careful in terms of the characterization per se. So to conclude my talk today, there is a considerable amount of development in the technology towards developing these maps, which has seen a very good amount of development. We have multiple cell types. We have a very focused technology, bioreactors, processing method, uh, you know uh, multiple categories of instrumentation which allow person to develop different kind of complex molecules, as well as a growing population which is understanding proteomics as well as genomics, which is also helping the monoclonal antibody development. There are 7 to 9 companies which are right now developing these biosimilars. They are not novel biologics, but definitely biosimilars. And also these biosimilars, India is actually leading a lot of biosimilar efforts. Again, these are antibodies, but these are much more economic than their original counterpart. More faster approval and advance in biosimilar development, I feel that will provide affordable manufacturing. Affordability is still a key. Still, a one vial will cost more than, in general, not less than 10,000 bucks. So if you go for from 10,000 to 85,000 to 90,000, this is a per dose, of any biologics molecule. So which is still very expensive, but there is still possibility that eventually these therapies will be much more affordable. Antibody fragments, which are the fragments actually shows the function. Antibody drug conjugates called ADCs, and gene therapy, which is CAR T therapy you have seen recently, are the some more advances which are in year to come. So biologics, as I said earlier, is going to take a huge amount of this is a huge opportunity for most of the students, specifically if this talk is this is the pharmacy student. They need to upgrade their skills. They need to understand this science much better sense, and if they could understand and work towards their skill set into this area, then there are very good opportunity which are waiting in those segments. So thank you everyone for your patient listening. This is my collaborators, my students who are actually helping us to, we run a program called Biosimilar Workshop. So these are our team which works for different. We also have a digital edition of Biosimilar Workshop, You can follow us on YouTube, Instagram, Facebook, and LinkedIn, where many of our talks are uploaded. You could understand, enhance your knowledge. These are some of our websites. Thank you very much.
Yeah. Thank you very much, sir, for your informative talk.
Yeah.
Your comprehensive presentation has really enlightened all of us uh, with basic understanding of monoclonal antibodies, the types of monoclonal antibodies, what are polyclonal and monoclonal antibodies. You also discuss the traditional approaches as well as the commercial approaches for the development of monoclonal antibodies in a very simplified manner. Definitely, the students have understood this process, critical process very easily. The way you presented the complete production line and the approval process is really very amazing. It was really amazing. Further, you also explained the various molecular and the functional analysis for the monoclonal antibodies, which will ultimately lead to the approval of these developed monoclonal antibodies. So it has definitely has added a great information to our knowledge, and I assure you that definitely all the participants will make the use of all these insights which have been gained through this particular session. Thank you very much, sir. Now, I
Thank
think
you.
let's take some of the questions from participants. Yeah, yeah,
Yeah,
Madhur,
yeah,
yeah.
yeah. Hi, Ratnesh. It was a brilliant presentation, very comprehensive. Though, I would like to apologize to you as well as to all the participants for 10 minutes of delay in starting today's session because of some internet issues. But you've still been able to manage to actually finish it well within the time. So thanks a lot for that. My first question would be that you said that while producing or while going for the manufacturing of monoclonal antibodies, there's a chance of viral contamination. So you need to apply either low pH conditions or and high temperature to actually take care of these viruses. But what is the impact of this on the stability of the antibodies?
So any low pH typically, so temperatures are not very well recommended though, but low pH is
Okay.
something which is, of course, is a routine practice. All the low pH have, some disadvantage on the total yield which is coming out for antibody, and that is also going to be. But at the same time, there is a risk which is involved with viruses. So of course, we do see drop in the total yield after
Okay.
viral inactivation, but it's not such a considerable loss that it cannot be compromised.
Okay, thanks for that. Uh, just wanted to ask you. I mean, I'm just diverting the topic from monoclonal antibodies to nanobodies now, because
Mhm.
that's something that we've been hearing about. Because nanobodies seem to be having advantage over monoclonal antibodies in terms of lower molecular weight. So,
Mhm.
is there any development happening in India on that part,
Uh,
like as
yeah.
in the field of nanobodies?
So I'm at least at the commercial level, I'm not at least aware that such development is happening. So
Okay.
certain European universities as well as certain companies in Europe and US both
Okay.
are working on nanobodies. Specifically, a couple of startups in Germany
Okay.
have also filed their patents as well as certain approvals process. So this is
Okay.
happening, but I'm not aware that any one of us, because our primary interest in country is developing more affordable therapies. So
Yeah.
since most of the antibodies which are now going off patent, there is a
Yeah.
huge amount of opportunity available, and that is also very fair and agreeable that companies are focusing on developing these affordable therapies.
Okay. And but then, do you see any advantage of nanobodies over antibodies, monoclonal
Of course,
antibodies?
yeah, of course, there would be advantages. Monoclonal antibodies are very complex structures. They are too big to handle. Any
Yeah.
nanobody means just part of the antibody. It's a very
Yeah.
small part of antibody actually, which is biologically active.
Right.
So that is definitely, but the science of nanobodies require its own time and validation. That is something which is a question at the moment. Antibodies are naturally produced, so the biological effector and functions are very well proven, and we are aware how it works. How it will happen in nanobodies and what would be the clinical performance end of the day, that is still a question. So, handling and engineering perspective, of course, it is going to be easy. Clinically, I'm not sure till the performance.
Yeah, thanks for that. The next question is like, you have actually explained the process of production in great details, but then this seems to be requiring a very, very state of the art and a very sophisticated setup to even probably do a R&D scale work. So what's the kind of setup that one would need to actually work in this area, even in the research phase? I mean, forget production.
Yeah. So I think I'll be answering question for two people. One, if considerably you are a student or an academic setting, so it's not too hard to work. You really don't need right now in current circumstances a very sophisticated infrastructure. Yeah, of course, in a industry you need a very sophisticated structure, and you don't need area. One 800 square foot is good enough for production of a millions of doses. That is not something which you need. What you need there is, specifically a cell type or even a E. coli, a simple E. coli which you have handled in microbiology lab. So the microbiology experiment, if you have ever done in your life, these biology experiments are not very far from there. So any E. coli can be simply replaced by a mammalian cell line, and one could do the experiment. Even if one can't do that and understand the protein, you can do by E. coli protein through any specific SDS page or a basic technique, so you understand the protein. Hello.
Yeah, Dr. Suvarna, would you like to ask any question?
Yes, sir. This is a specifically question from my side itself. Like you had such a huge presentation of monoclonal antibodies. Can you just throw some idea on the in the current scenario of this pandemic, corona pandemic, how this development of monoclonal antibodies is going to be very helpful in this situation?
Yeah, so therapies are already started, worldwide in our country. In our country, Bharat Biotech, which is a Hyderabad-based company, along with certain partners, have started the development for monoclonal antibodies. Therapy for viral infection is something which is going to be, if it is possible, it is going to be very successful. But again, as you see the timeline, specifically the developmental timeline and getting maps, see all the recovered patients are having antibody. So antibody is still there, but commercially manufacturing it and then giving to patient is a very different ball game altogether. So it requires typically a 3 to 5 years of timeline if everything goes very well. So Bharat Biotech is still right now uh, is leading the consortium which is developing antibodies in our country. There are a couple of more companies at the private scale which have also started in this area.
Okay.
So I have one question came up. I'm sorry, I'll answer. So one of the question which came from
Yes,
Patra,
sir. Yeah. Yes, sir.
that please share insight with respect to computational approaches for the static
Right, sir. Right.
antibodies with modulated function. So computational approaches are also being specifically, these are in silico methods. These in silico methods by different bioinformatics. So since we have sequences almost online, so genetic libraries, all the genomic servers are carrying almost entire sequence of human genome is already with us. Indian human genome is also established. What it allows us to basically take all the sequences online, and one could actually design the most, the best possible. So majority of the antibodies fails in their primary biological functions. So if that biological function is somehow can be optimized through computational approach, that the design is appropriate, then we would be able to deliver the or I would develop the antibody in a much better sense. So yeah, in silico methods are taking a great amount of interest at this moment, but still, I can't compare with the regular drug design. So like CAD approach, which is for drug, is there, and the development which is around that CAD approach in a synthetic drug. So for antibody, one has to still reach to that position, but a lot of companies are utilizing those approaches. Yeah.
Yeah. Ma'am?
Just
Sorry.
one question that I had was, you explained very well what are monoclonal antibodies and what are polyclonal antibodies. Do you see any application of polyclonal antibodies for treatment of viral infections? I mean, why am I asking this? Because viruses can actually mutate. And if you have polyclonal antibodies, I mean, there could be a better treatment with polyclonal, because monoclonal probably may not find the correct fit on the mutated virus.
Yeah, so it would be very difficult because polyclonal is not necessarily, it is going to create a severe side effect. If an antibody is not appropriately fitted into the target antigen, then the polyclonal is good for diagnostic purpose.
Hello.
But whenever it, hello, so the
Yeah, hello.
polyclonal, yeah, am I audible?
Yeah.
Yeah, so polyclonals are good for diagnostic purpose, but whenever one has to develop a therapy, unfortunately, it cannot be polyclonal. Considering the multiple epitope, it can bind and create a severe immune response. So to control the immune response and side effect, one has to make sure that it is focused and targeted, which is monoclonal.
Right. Okay. And in terms of cost, because you've already mentioned that a single vial of an injection would cost around minimum 10,000 rupees, or I mean, and it can go as high as a lakh of rupees, which is still
Yeah.
an unaffordable thing for a typical Indian. So,
Mhm.
is there any initiative by government or any such activities going on to reduce the cost?
So government is funding certain projects uh, to these companies which are producing these biosimilar molecule, where the target is to reduce cost of therapy as low as 5,000 a vial to 4,000 a vial. That is the only way government is right now. The government which should come, and which we have recently done even a discussion in that area, whether insurance sector can reduce the price. So unfortunately, we don't have a policy where it says that government will subsidize the cost of certain medicines. So we don't have, unfortunately, such a situation through government. So the only way is they are funding and making more competitive. The Indian companies are more competitive, and so Intas has recently launched a drug for something like 4,000, 5,000 a vial, but that is the least which you can hear, in the market. Not more than, I mean, not
Yeah.
more than that.
But I think something should be done, right, to reduce the
Yeah,
costs further. But I mean, of course, one can understand that there are so many nitty gritties, and it's so complex to produce these
yeah,
monoclonal antibodies. But
mm,
still, something needs to be done in this field. Yeah.
yeah.
So thanks a lot, Dr. Ratnesh, for patiently answering all our questions. And I would now like to invite Dr. Vandana Patravale, your guide, your mentor, to the studio, and to tell all the participants. I think we've been probably telling during every session earlier that out of 4 speakers that we had for this international webinar series, 3 are Vandana madam's
Yeah.
student. So we requested Vandana ma'am to please join this webinar today, at least the concluding session, and say a few words about your students and even about the webinar series. Yeah, ma'am, please.
Thank you so much. At the outset, of course, my compliments as well as congratulations to Indira College of Pharmacy Pune team for hosting this very good international webinar series on biologics, the future of therapeutics and healthcare management. I would summarize it in 5 E's: excellent theme, excellent teamwork, excellent response of participants, of course, excellent take home value, and excellent speakers. So, speakers, be it on aptamers by Dr. Teja, who came from USA, or enhancing efficiency and biologics through use of adjuvants by Yogesh, modalities of delivering genes, Dr. Abhijit Date, and today we heard monoclonal antibodies by Dr. Ratnesh. As you already said, 3 out of 4 were my students, and that itself says a lot. I'm really, really proud of each of them. And as you know, for teacher, what more can be better than listening to her or his own students excelling? Really good. Now, in fact, all the speakers made it actually very, very simplified for all the participants to grasp. And that is something which needs to be appreciated a lot, because there are participants who are students, who are teachers, who are novice in this area like me. So it really became very important for us to listen to them. Now, since Dr. Madhur said that I should be talking something about, how Indian companies as well as academia are catering to this challenge of working in biologics, I do not know more. How many of you know that Government of India took this cognizance only in 1980? So it is only 20 years since then, 1986, DBT got established. And if you look at DBT website today, it has sponsored over 5,000 projects, of which 94 are international projects. There are about 5,000 scientists who have been supported, and about 286 technologies which have got developed through this initiative. Besides this, there are about 19 autonomous institutes and public sector undertakings which have got established. And in fact, if I have to give you examples, it's like TIFR in Mumbai and NCCS, which is there in Pune itself, comes under this autonomous institute. Besides this, there were efforts to bring together different regions, and in region wise, there's a Pune cluster wherein NCCS is collaborating with ICER Pune. You would be shocked to hear, if you do not know already, that they got grant to the tune of 125 crore to establish this cluster. And they have been working on model organisms to human diseases. So likewise, there are other ones which are in Bangalore, in Calcutta. Of course, not to forget a very good work which ICT has been doing, though it is not coming in clusters. On its own, ICT has established DBT ICT Center, which focuses on biofuels, and Dr. Ratnesh and Dr. Prajakta Dandikar Jain, who have been working really very jealously, very enthusiastically to see that we are in the forefront. Industry-wise, you do have Dr. Reddy's, Biocon, Zydus, Cadila Pharma, as well as, of course, in Pune itself, Serum Institute of India, is working really very well along with Bharat Biotech. So just to give you an idea, which are those areas? No, sometimes people ask me, which are those areas where we as research scientists can work? And DBT has given those priority areas very well. And they include bioinformatics, they include biofuels, bioenergy, biotransformation, biosafety, medical biotechnology, and of course, genomics, if you really understand that very well. So we do fall under life sciences, and therefore we can submit any project which falls into these thrust areas to DBT. If you consider diagnostics, it could be ELISA, it could be lateral flow, could be molecular diagnostics themselves, PCR like techniques which you are seeing now, or it could be as simple as colorimetric assays which are being developed, or fluorimetric assays which are being developed to diagnose diseases. On vaccine front, one could propose a novel vaccine platform. It could be novel route, could be novel vaccine adjuvant, could be DNA, RNA, virus-based vaccines, even non-viral based nano carriers. And as far as novel therapeutics is concerned, it could be gene delivery, any of the biologics, could be monoclonal antibodies, also novel methods of beta cell isolation fall in this category. So you would see Indira College of Pharmacy actually has touched upon one from each area. So we heard about aptamers, which can be used both in diagnosis as well as vaccines. We heard about gene delivery, which falls in therapy. We also heard today about monoclonal antibodies, which falls in this therapeutics class. So I would say that all in all, no, believe me, nothing is impossible. Once we decide, we have to move forward, and it offers plethora of opportunities to excel. If you just look at even COVID, you know how many projects DBT sponsored? 34 in diagnosis, about more than 10 vaccine platforms. In fact, one of our project on mucosal vaccines got approved through this route. 14 on preventable technologies or interventions, 10 therapies, and 2 for drug repurposing. So I would just say, do not give up easily. As Rabindranath Tagore says, if I can't go through one door, I'll go through another. And if not, I'll create another door. And that's what Dr. Ratnesh Jain has done it here at ICT. So once again, heartiest congratulations to this team, ICP, including Dr. Anagha, Dr. Madhur, Dr. Kannur, and all the organizing team members for doing excellent work. I would say keep doing your best, and best always will be yours. Thank you so much for giving me this opportunity.
Thank you so much, ma'am,
Thank you so much,
for
ma'am.
your inspiring speech. You've always been an inspiration to all of
To
us.
us.
All of us.
Absolutely true.
Thank
Absolutely
you.
true.
Thank you so
Yes.
much. My pleasure
And
always.
And today I must say that I must be doing the last part, that as we conclude this international webinar today, I'm very happy and I'm happy to express my gratitude towards all the four speakers, Dr. Teja, Dr. Abhijit, Dr. Yogeshwar, and Dr. Ratnesh Jain, and to you, ma'am, for accepting our request to give concluding remarks. And your words of encouragement means a lot to us.
Thank you.
And yes, as you said, that when one door gets closed, you can open another one or create another one. Though we are in very nascent stage, but definitely yes, we will walk on the path which you have showed to us. And I'm also thankful to all the viewers, like all the videos were viewed by more than 1,000, 1,200 viewers. And at a time watched by online around 200 to 50 people with very good questions, like those who are interested. So I'm really very thankful to all of them. And most important thing, I'm always proud of my team, ICP, who is always, I should appreciate and applaud their efforts in bringing all this, because
They
we are
are
not
good.
technocrats. We are pharmacists, but they are, I must say, they are technocrats while arranging all these sessions online. So, uh, all the viewers, please, like many a times, what happens is we listen to something, we get encouraged, and then we leave it because we get busy in our day to day work. But please carry a thread of this webinar as a take home. And if we could do at least a percent of it as our contribution towards the research, I think this is the best outcome. This will be the best outcome of the seminar. So thank you, Vandana madam. Thanks a lot, and thanks all the speakers and my own team for doing this.
Ma'am, it has you have been our great motivation and great support. Without you, it would have not
Yes,
been
ma'am.
possible for us
Yes.
to do anything. Yeah.
Right,
So,
right,
and
sir.
Vandana ma'am, thanks a lot for being here. Dr.
Thank
Ratnesh,
you.
thanks a lot.
Thank
Thank
Yes,
you so
yes.
much.
It was
Yes,
such a wonderful, simplified yet very
Always
informative
my
session, Dr.
pleasure.
Ratnesh. Yes.
yes.
Thanks. So
Uh,
I think we will meet on some other similar another platform with some similar other topic of interest very soon.
yes.
Thank you,
Thanks,
ma'am.
everybody.
Thank you.
Thank you,
Thank
Thank
you.
everybody.
I
you.
request
Thank
Vandana
you,
ma'am and Ratnesh
Ratnesh,
sir to be
for
in the
making me
studio.
feel proud.
Yeah, thank you.
I request Ratnesh sir and Vandana ma'am to be in the studio. We will be going off the broadcast. Thank you, participants. Thanks, one and all. Thank you. The broadcast is being ended.
Okay, thank you.